Welcome everyone to our June meeting of the ReproNim webinar. We are so honored today to have Hugh Garavan with us. As way of background, Hugh received his PhD in cognitive psychology and behavioral neuroscience from Bowling Green State University in Ohio. He then completed his postdoctoral fellowships at Cornell University and the Medical College of Wisconsin, and was an associate professor of psychology at Trinity College, Dublin. He moved back to the US to the University of Vermont in 2011. His research uses structural and functional neuroimaging to study cognitive control and reward processes with a particular focus on adolescent development, addiction, and related mental health issues. He's been part of many large-scale projects, including Imagen, ABCD, HBCD. He runs a number of training efforts focused on complex systems methodologies for large-scale neuroimaging datasets, and is co-founder of the Enigma Addiction Working Group. Many of those initiatives I've just mentioned are all part of the greater ReproNim family, and it's because of that it's such a pleasure to have Hugh speak today. So I'll turn the floor over to you, Brother Hugh.
Brother Hugh, no, that's
Part of the
excellent.
family.
All right, well, thanks, David. Thanks for inviting me. Thanks everybody for being here on a Friday afternoon. I don't know what it's like where you're at, but it's a nice sunny Friday here. Um, okay, so it's a bit of a hodgepodge. I'm going to talk about, as you see in the title, both individual differences and then also a particular focus on cannabis use. The topic here is going to be the ABCD study. See if I can get my things to work. Excellent. So I know some of us, me, live and breathe ABCD, but not all of you do. So just very, very briefly by way of background, this is a large, ongoing, longitudinal study, almost 12,000 kids. There's over 2,000 twins by design, half of them monozygotic. We're following these kids. They all started back in 2016. We've been following them for about 10 years. We hope to keep following them. Lots and lots and lots of assessments, imaging, genetics, psychometrics, hormones, mental health, cognition, environment, personality, biosamples, geocoding, and all of the data are made publicly available. So I guess a little bit part of today is not exactly a sales pitch, but I'm just going to try to give you a feel for the different things that are ongoing with this study and make the case that actually this is a really good time to start working with these data because it's starting to get very, very rich now with lots of longitudinal data available. Like I said, it's been going for a while. So we started back in September of 2016. It took about 2 years to, accumulate 12,000 the baseline sample, and then there's another assessment 6 months later that's shorter, a bigger one a year later. That kind of has everything as same as what the baseline had, but without the imaging, an 18-month assessment, and then a 2-year repeat of the baseline. And so this is the data collection. You'll see there's a few, wobbles, COVID, things like that happened. But, it's gone really well. You'll see there's over about 160,000 assessments and up to 50,000 biennial MRI visits. Now, that number is a bit of an exaggeration in that, people miss their visits or because of braces can't get in the scanner. So we don't actually have 50,000, but we have tens of thousands of data sets. So it's a very, very rich data set. A lot of time and effort was put in trying to make it as, as diverse as possible. But I do want to flag for you that, it's not perfectly representative. It's very hard to do anything perfectly representative. And there is this issue that, there's missing data and that tends not to be, equal across socio-demographic groups. So just kind of a caveat. So you can see here, these are the sort of metrics we plot. So the year six data, for instance, that was, we completed a year or so ago. There's nice representation across, family income levels, but you'll see that the missingness tends to be most exaggerated in the lowest income. So just that kind of caveat and qualifier that the data set, while rich and diverse, is obviously, imperfect. Okay, so what am I going to talk about today? So individual differences, like I say, a bit of a hodgepodge to give you a feel for a few different things that are ongoing, kind of give you a sense of what can be done. I don't think I'll get into anything in any great detail. And then we'll talk a little bit about cannabis. So primarily the focus here, the ABCD study primarily designed with a focus on individual differences, right? So the goal is to explore what might influence development trajectories. You know, I think it's a really interesting idea. It's an interesting type of study to do. The changes between a 9-year-old and a 20-year-old are pretty huge. And as you know, a lot of mental health issues emerge during that period. And a lot of kind of personalities and kind of life trajectories don't get established, but get, heavily influenced. We know that a whole bunch of things influence development, sleep, physical activity, intellectual activity, sports, and so on and so forth. But we don't really have good quantification of, how big those influences are and how they might interact with one another. So, like I said, the study itself is getting more and more interesting now as we are accumulating more and more longitudinal data. And this is where we start to really get to ask super duper interesting questions. So, for example, in this 12,000, you're going to have, if your interests, for example, I don't know what all your interests are, but many people are interested in mental health issues. Of this 12,000, you're going to have significant numbers of kids develop anxiety disorders, depression, suicidality, psychosis, substance use, and what have you. Once those things start to emerge, and sorry, not to keep focusing on the negative, but you'll also have kids who will absolutely thrive and will do really, really well. Once we have those outcomes, how interesting to be able to go back and look at their early, their late childhood, their developmental trajectories prior to these outcomes. Can we get insights into what might have predicted these miscellaneous outcomes? Was for those kids who develop psychosis, is there a signal to be had in their genetics, in their brain, in miscellaneous peer influences, family histories back at ages 9, 10, 11, and 12 that could help us, retrospectively predict their outcomes? So it's, quite an interesting set of research questions that we can ask. So what I'm going to do though is focus on one part of it. The task fMRI, we'll talk a little bit about the opportunities with it, some of the challenges. I'm going to touch on just a few of these issues: longitudinal trajectories, computational modeling, neural signatures, neurotypes. A couple of these topics have been done by Tony, who's with us here. So if you've got laudatory things to say, I'll take them. If you've got challenging questions, we'll pass over to Tony for all the tough ones. And then because a lot of the focus here, the study originated out of the National Institute on Drug Abuse, we'll talk about kind of one of these clinically relevant questions to do with cannabis use. And I will try to stay cognizant of the time. Okay, part one. Let's talk about individual differences and task fMRI. So my background is cognitive psychology. I came to brain imaging with an interest in task fMRI, challenging the brain with, with cognitive probes, seeing what sort of brain regions show what sort of functional responses. Of late, though, somehow task fMRI has become the ugly red-headed stepchild of the neuroimaging world. People tend to like brain structure and resting state connectivity, not so much task data. And this disturbs me, especially the ugly redheaded step, because I do come from a country where we have lots of redheads. So I like redheads. I like task fMRI. So part of today is going to be talking a little bit in defense of task fMRI, which has its challenges, but uh, I'm I've just I've noticed over the years how uh, fewer and fewer people are working on task data than working on some of the other MRI modalities. Within the ABCD study, I don't think I need to go into these in great detail because it's not pertinent. But just to let you know, we have three tasks that we use. There's sort of a reward task. You get a queue, and if you respond fast enough, you can win $5 or you can win 20 cents. Or if you respond fast enough, you can avoid losing $5 or losing 20 cents. So we can look at kind of reward anticipation, reward outcome. There's a response inhibition task. So stimuli come up on screen. You've got to respond as quickly as you can. On occasion, when you're about to make your response, you get another signal saying, stop, abort, don't push the button. So we kind of look at that response, those response inhibition processes. And then there's an end back task where there's stimuli, and you have to remember the sequence of stimuli and remember if the one on screen is the same as two back in the sequence, right? And then there's a control condition where you're just looking for a particular stimulus, a sort of zero back. So again, trying to probe working memory abilities. So, we're hitting reward processes, reward anticipation, reward outcome, response inhibition, sort of executive functioning processes, and then these working memory processes. And that constitutes the task fMRI battery. Like I said, now is a great time to be studying longitudinal effects in ABCD. And actually, this is out of date, I'm sorry to say. So we have data releases every year. These are data from the 6.0 release, but since then we've had the 7.0 release. So lots more data in older participants. But even in the 6.0, you can see here we have data from kids ages 9 right up to just shy of 18. And now we're well into the 18s in the 7.0. So you have a full 9 years of development. So we can look at things like just the, brain structural development, cortical, in this case, cortical thinning, cortical maturation. Um, you can, do the same thing, but, looking at surface area. Surface area has, tends to follow different time courses. Cortical thickness tends to just show, for the most part, linear decreases. Surface area shows a more varied pattern of change across the brain. Subcortical volumes also show a more varied pattern of change across the brain. So you can already see where I'm potentially going with this. If we can quantify brain changes, for individuals, now we can start to look at, well, what might have influenced those trajectories? If you're engaging in lots of physical activity during your adolescent years, how does that impact on your brain trajectory, your brain development? If you start using substances at age 14, does that impact? If you're doing a lot of screen media activity, what sort of brain changes might that make to you? So now that we can have these trajectories and we've got good behavior to go with it, we can start asking a bunch of interesting questions. We can also then ask the same questions, but not brain structure, but brain function. And again, brain function changes considerably, much more varied than the cortical thinning. So some brain regions activity across from 9 to 18 is going down. In some regions, it's going up. In other regions, it's got these interesting nonlinear patterns. This is all very interesting to see how brains mature and develop. We can start looking to see, well, what sort of brain regions show similar developmental trajectories. So we can look across the brain and look at different clusters. So, the dominant pattern here is just this single, essentially linear decrease. But if you look for a two-cluster solution, you'll see some brain regions are going down, some are going up. A three-cluster solution, four-cluster, five-cluster. So, you can see what sort of brain regions are showing what patterns of neurodevelopmental change. And all of this actually will be available as an online tool that you can actually explore voxel-wise, what sort of brain changes are happening. You can also, surprise, surprise, look at behavior, performance. So in the stop task, for instance, you get this measure, the SSRT, kind of a measure of how efficient, how good you are at response inhibition. It might be kind of hard to see, but there's a nice linear decrease. People get faster and faster at that over time. On that end-back task, the working memory task, you can measure performance and you can see that it increases over time. So, cognitive abilities, executive functions are improving over time. We can do things like look at clustering of the performance. So you see, this is just a simple three-cluster solution. This group in yellow at baseline, so ages 9 and 10, start off, relatively poor on the task. They get better, but, not hugely. And the second group also starts off relatively poor, but they get noticeably better. And then a third group that starts off pretty high and, they get better too. So, we can identify people who vary in how much cognitive development improvement they show over this age period. And then, of course, we can see, well, what brain regions map on to those different types of clusters, those different groupings. What's the pattern of someone who goes from being not really great at working memory to be really good at it? What brain changes are accompanying that? And then again, same thing. What are all the things that might influence that? Is it nutrition, et cetera? Is it genetics, and so on and so forth? All these questions now become very tractable. Right. We have a boatload of assessments in our participants. So there's a boatload of cognitive measures. We have those developmental trajectories as well. This is just, other cognitive measures we can bin by, when the scans happened. We have mental health measures, very extensive assessments. So again, we have all of these measurements and all of these trajectories waiting now to just be put together to try to make sense of, what are the brain correlates of this, if you will, normative decrease in externalizing symptoms, and so on and so forth. And of course, in these trajectories, there's lots of individual differences and lots of individual variation, and therein lies some of the interesting research questions we can ask. That's a complicated slide, my apologies, but just to flag for you, something quite interesting that we can do now. So this is, for example, a project that we're hopefully going to be finishing up and submitting for publication within a couple of weeks. What people are interested, you've probably heard this, there's an awful lot of anxiety these days about screen media activity and how that might impact on mental health or indeed on brain. So what we can do with multiple measurements, and we have measurements of this every year or every 2 years, we can track, how much screen time someone is engaging in, what their mental health scores are at ages 9 and 10 and 11 and 12 and 13 and 14 and 15. We might imagine that screen media activity at one age is probably predictive of screen media activity at the next age. We might imagine that mental health measures at one age are predictive of mental health measures at the next age. And that's kind of what's captured by these patterns. But the critical issue that we can ask is the kind of cross-lag. Does screen media activity at this age predict future mental health? Or does mental health at this age predict future screen media activity? So as best we can with human correlational data, try to get a sense of what's the temporal sequencing here. What tends to precede what? Because we know that there's strong correlations between the two, but we really don't know if one is causing the other and what that directionality. And this type of analysis gives you an insight into it. And just to add to the fun, we've done this now looking at discordant twins, which is even a more powerful approach towards causal inference. So instead of just looking at individuals and how they change in screen media activity and mental health, we can look at how a pair of twins differ. And the beauty of that then, so we can see if the difference in how much screen media activity between a pair of identical twins and how that might be related to a difference in mental health between those identical twins by using the monozygotic twins, to a large extent, you can control for environmental factors and genetic factors. So it helps you maybe zero in on what might be a causal influence. So again, all these types of analyses, because this is screen time and mental health, but start to imagine research questions of your own. Maybe it's brain and cognition, maybe it's physical activity and who knows what, and mental health scores and what, and so on and so forth. Or maybe it's environmental circumstances and cognition. Maybe it's peer influences and how that might change over time, and substance use. So, once you have this framework and you have these data, all of these sort of questions start to become tractable. Okay. One of the issues, I won't belabor this. I know obviously this is the bread and butter of ReproNim, right? These whole reproducibility issues. As you know, there's a lot of anxiety that there are very small effects and very small samples in our that we work with. I won't belabor this. But what I want to point out is that one thing we've learned so far, and I guess this is a bit of a sales pitch for studies as large as ABCD and for kind of data pooling endeavors, is that it seems to be the case that true effects tend to be small effects. So this is a project done a couple of years ago by a postdoc here. And, there's no brain imaging measures here. So what Max did is he just looked across about 200 measures. All of them actually plausibly could be related to one another. And he just essentially correlated everything with everything to just get a feel for what sort of correlations and the correlation strengths might be. So some correlations are pretty high. I think this was all done with the baseline data, just when the kids were ages 9 and 10. So as you might imagine, sleep and a score of just total mental health problems, pretty highly correlated, 0.58. age and pubertal development, maybe not as highly correlated as you expect, but correlated 0.17. Amount of time spent, on screens and your weight positively correlated. Other things like, sleep problems and total IQ, very weakly correlated. So he just kind of looked at all of these correlation matrices, put it all together, and like essentially this is what the distribution of correlations looks like. So, and a lot of those correlations, like again, if you start to match them, they all kind of plausibly could be related to one another. But the median correlation was 0.03. Okay. So, and I skipped the slide that kind of talks a little bit about, the inflated estimates that are in the literature. I can come back to that if anybody wants me to elaborate. But again, I think this is sort of your bread and butter, all these issues. But the average correlation here, the median correlation was about 0.3, even like the 90th percentile was 0.17. Typically, effects, the correlations are bigger if you're within versus between instruments, within versus between a domain, if you're within versus between a reporter, but they're not hugely different. And even if you start to apply, better statistical practice, adding nuisance covariates, applying statistical threshold, statistical corrections, it doesn't make a huge difference. It's a bit small, but if you can read it there, essentially, even if you bond for only correct and you add, your kind of standard complement of nuisance covariates, your median correlation is still just 0.09. So to be honest, for the most part, if we see correlations in the literature in our lab that are, 0.5, 0.6, 0.7, we almost automatically just don't believe them, right? It's just not our experience. The true correlations we suspect are always going to be much smaller. Just our literature has lots of, spuriously high correlations. Another big issue, obviously, with the task fMRI data is reliability. This is a bit defensive. Let's just skip this. But reliability is clearly an issue with the task data. One of the real challenges for relating a task activation map to some individual difference in cognitive abilities or mental health is that the task activation measure might just not be that reliable, and that really creates problems. And that was the whole Marek paper that I'm sure you all know lots about, showing that these certainly using kind of standard mass univariate approaches show very weak correlations, and you need thousands of participants to really unearth them. Multivariate approaches do seem to be a lot better. This is work that Tony has led, and hopefully we'll have a paper out on this very, very shortly. Essentially, without getting into the details, but we happily will if you like, you can come up with sort of an optimal classifier. In this case, let's say the two-back task. You recall I said there's a working memory condition where you've got to remember if from the sequence of stimuli, if the one that's currently on screen is the same as two back, so you've got to keep a running memory. First is just you're looking for a target stimulus, so the much lower memory. You can try to create a classifier that will best discriminate your brain when it's doing the more demanding task versus the less demanding task. And if you do this in a multivariate way, you can come up with a pretty good way of classifying whether your brain is doing the more demanding or the less demanding. So these are area under the receiver operating characteristic curve, looking at our ability to separate brains, whether you're doing two back versus zero back, for the baseline data for year 2, for year 6, year 4, rather, year 6. And you see it's pretty good. So, there's clearly a signature that's quite consistent across people of, when your brain is engaging in a more demanding task. These are the brain regions that are contributing to that classification. These are essentially the standard regions that we would anticipate to be involved in working memory, a lot of frontoparietal cortex. I like including this. I don't know if I don't think Tony gets as much of a chuckle out of these results as I do, but you can, another contrast in this task, it's doing a working memory task, but sometimes the stimuli are faces and sometimes the stimuli are places. And can we build a classifier that can distinguish if your brain is currently working with face stimuli or place stimuli? And the answer is absolutely you can, almost like with 100% accuracy. This is across all people at all time points. And the reason is that I think there's just a very robust signal in your brain when you are viewing faces, this fusiform face activity. So, with multivariate analysis, we really can discriminate very, very effectively different task conditions. But the primary reason for telling you all of this is that, well, two things. It nicely reduces brain activity to just a single score. This neural signature kind of is a measure of the whole multivariate pattern of activation. We can then test to see, does that change with age? It does. Um, we can also relate it to a whole bunch of, phenotypic measures. This is a bit busy, but basically what you're seeing just up here, the distribution is just the correlation between this measure. In this case, it's a CBCL, a mental health measure, and each individual ROI. And then the red dot is just the correlation with the neural signature. And for the most part, you can see that the neural signature does better, shows a stronger correlation than the bulk of the individual regions of interest. Um, but when we look at the reliability, which I think is maybe the most interesting, again, if we just want to look over here, so this is the reliability. These tasks are done in two halves. There's a first run and then a break and then a second run. So what you can do is you can quantify the reliability of activation, the ICC from run one to run two. If we do that for each individual ROI, you get this distribution. If you do it for the neural signature, you get this data point. So these reliabilities are not great. Again, this is 0 here. This is 0.1. But at least for the end-back task, the neural signature's reliability is starting to be in a pretty acceptable range, 0.5. And this is true for baseline, year 2, year 4, year 6. You can see that the reliabilities of the individual ROIs is creeping up as the kids get older. But the neural signature, just this multivariate measure, is much more reliable. So therefore, it has great potential for being a better measure of behavior, mental health, things that you might want to correlate with brain. Okay. Again, I hope I'm not overwhelming you by moving too quickly. The idea is just to kind of give you a flavor for the sorts of things that we're doing with these data. Another thing we can do for reliability, and again, I'll go through this a little bit quickly, just because again, this gets complicated. We can get into the weeds. This is also work done by Tony. Often when people talk about brain behavior associations and we need to optimize how do we improve the reliability? How do we improve the strength of the brain behavior? A lot of the emphasis goes on the brain, but also we can extract better behavioral measures. So from the stop task, you can get like a nice performance measure. This SSRT, it measures the speed, the efficiency of response inhibition. But you can also apply these kind of cognitive process or computation models to decompose the task into more kind of elementary psychological functions. So there's evidence that's accumulating, there's a decision to be made, there's a decision threshold. There are specific processes involved in going, specific processes involved in stopping, and so on and so forth. And what you can do is, like I say, extract these hypothesized parameters that underlie task performance, and then we can look at them and their relationship to brain, for instance. There's a few here that are of particular interest. Evidence accumulation, so the efficiency by which you accumulate goal relevant information to help make your decisions. There's actually, I won't belabor it. There's a few other ones but we can just kind of skip those for now. But the point is, and again, this is a busy slide, but I'll just kind of guide your eyes over here. If we try to relate, let me guide your eyes over here. This is our standard SSRT measure, and we can look at brain activation associated with it, and it does pretty good. But one of these parameters, that evidence accumulation parameter that we get from this cognitive process model, this action is all done in a cross-validation. So out of sample, the association between this parameter and task activation, again, a multivariate analysis, it's explaining about 27%. So the brain data is explaining about 27% of the variation across individuals in this parameter. And 27%, just for it to be said, is pretty good. Again, I'm assuming all of you know the Marek paper. I mean, you do a mass univariate, your single best voxel might explain 1% of the variance. So, being able to get a boost this big, I think is interesting. And again, it just underscores that there is there are rich, insightful data to be had from these task data if we do the brain analysis well and also we do the behavioral analysis well. Okay, one more thing to talk about this. Another approach to this, again, the focus here is just trying to maximize these brain behavior associations, is that you can, instead of treating your entire sample of 12,000 kids as one homogeneous group, is just try to embrace the fact that there's probably heterogeneity in here. People probably use their brains in different ways to achieve the same end goal. So what we can do is if we're trying to relate a behavioral measure to a brain measure, maybe that's going to be different across these different clusterings of individuals. So the strategy and approach like this is first, let's see if we can identify the subtypes. So again, we can talk about the details, but this is just basically looking at the patterns of ROI activation and using some of these clustering techniques. And in this case, it kind of found that there's three separate groups. Again, with a certain threshold, you can identify three groups that seem to use whose pattern of brain activation are different from one another in how they achieve the task. And then what we can do is we can look at the brain behavior associations separately for each group. So if group A uses their brain differently than group B, we might expect that if we lump them together, that's going to lower the correlation between brain and behavior. Whereas if we look at those groups separately, we might get a better correlation within each group. I hope that makes sense. And, basically, this is, the proof of concept type of analysis. This hasn't been published. But just to kind of to cut to the chase, we do see that with this type of algorithm, with certain thresholding, we get a three-group solution. When we look at all the groups combined and we try to correlate brain activation with performance, we explain about 15% of the variance. When you do the same analysis separately for each group, you get a boost. So attending to the heterogeneity, identifying subtypes or neurotypes, if you will, and then doing your analysis separately for those subtypes, you get stronger brain behavior associations. So trying to accommodate that heterogeneity. So point is, there's a lot of different strategies that we can employ to try to boost these brain behavior associations, and it's a big focus of the group. Okay, don't worry, we're more than halfway through, but deep breath because we're going to take a bit of a right turn here. Okay, so that's kind of background of what we're doing on the specific set of questions to do with a task. Cannabis use. So let's talk about that because, as I mentioned, the ABCD study in part is very broad in its focus, but it started off with a primary interest in trying to understand what things like substance use might do to the developing brain. And in one slide, I'll try to give you the background, but I think this is probably going to be pretty obvious to all of you. So we know that there's substantial brain changes that happen during this adolescent period. We know that, I'm sure this map is out of date, but that, cannabis is becoming decriminalized, legal all over the country, all over the world. We know that cannabis is getting stronger. So the ratio of THC to cannabidiol has increased substantially in the last number of years. We know that it's still very popular. Cannabis is still very, very popular amongst adolescents. And we know that the perception of its risk has gone down over the years. And we also know that, with this brain development, that the cannabinoid system itself changes during the adolescent years. There's more CB1 RNA expression in late childhood and in adolescence than there is in adulthood. So this kind of raises this whole set of issues. Crikey, is the presumed increased availability or decreased risk perception of cannabis and the increasing potency of cannabis, is this a matter of concern? Does it have an effect on the developing brain? And that's kind of one of the questions that we set out to address. There's been a kind of program of research in the group for the last while. And again, Tony is actually spearheading this project as well. So, just a quick aside, doing this stuff is tough, right? I mean, human research is inherently correlational, right? And even if we have the nice temporal information from ABCD and large samples and all the things, it's still always really, really hard to disentangle, what might be causing what. So you might find an association between brain and, let's say, cannabis use, but concluding that one is a consequence of the other is much, much more challenging. Um, I won't belabor this. I'm not saying that big data are necessarily the solution, but I think it certainly helps when you're dealing with small effects, when you're dealing with, interactions and stratification, because maybe cannabis has an effect, but really it only has an effect in those individuals who are also using alcohol, or in those individuals who've got a genetic risk for it, or have some sort of comorbid anxiety disorders. Um, when you want to try to deal with all the confounding that inevitably accompanies human research like this, or you want to build in a proper replication, you really can only do this, I think, with large, large samples. So I think these are the sorts of questions that a study like ABCD is optimally designed to address. Um, so there's lots that we can do with cannabis in adolescence. So we can look at exposure effects. I'll just kind of walk through a few of this. So again, these are data that Tony has wrangled. This is timeline follow back. So essentially each row here is a participant, each dot is a cannabis use experience. So now we can kind of properly quantify how much and when cannabis our participants have had. We can quantify that as this. This is like at the age at which participants started using cannabis. You'll see that it ramps up. We're accumulating more and more users as they get older. The vertical lines here tell us when we have our assessments, including our brain imaging. So it lets us answer, this kind of guides us as to what sort of research questions we can do. We can do this for cannabis. Tony has also done the exact same for alcohol and for nicotine. So we can have like really good quantification now of when the kids started using, what they're using, and then use that to guide our research questions. We can do things like look at latent classes. So this is just looking at the groups of individuals who started using cannabis and how much cannabis did they use. And you'll see for the most part, we have these different groups who start using, and once they start using, they keep using. Tony, I've been meaning to come back to you now that we have the 7.0 data. This blue group is really interesting, huh? So they started using, they kept using, and then for whatever reason, they stopped using, or at least they slowed down their use. So if we have lots of those kids, isn't that an interesting group to look at and to compare them to the kids who started using, but then just kind of continued using and escalated use? Like what's different between those two? What sort of brain correlates might there be of those types of behaviors? So this is one type of analysis we can do. We can look at one of the best predictors of adolescent cannabis use is prenatal cannabis exposure. And there might be lots of different reasons for that if you stop and think about it. But just to, this slide is a bit busy, but just to let you know that what is just shown here is essentially what are the odds that if you're using cannabis during adolescence, that you had a prenatal exposure to cannabis. To quantify that a little bit, if you are using cannabis, there's like an 11% chance. If you're using cannabis as an adolescent, there's an 11% chance within our data that there was also prenatal cannabis exposure. If you're not using cannabis as an adolescent, it's more like 5%. And we can also look at that across ages. So just to unpack that a little bit, the younger you are as an adolescent cannabis user, 12 versus 14 versus 16, the more likely you were to have had prenatal cannabis exposure. So, how interesting once these numbers, the absolute numbers increase to see, what might be, if you will, apologies for causal language, but what might be the effect of prenatal cannabis exposure, at least the correlate of prenatal cannabis exposure on what your own cannabis use as a teenager might do. There have been a few papers already published on this, obviously, in the ABCD study. This is a really simple, and I say this as a compliment, paper. Essentially, what they did is they exploited the longitudinal data. So essentially, they looked at 46 variables acquired at age 10 in kids who were cannabis naive. They then identified the kids who went on to use cannabis, and they said, well, were there any differences at age 10? Really, really simple. And of the 46 that they looked at, 29 showed significant differences. So those kids who went on to use cannabis, differed in prenatal cannabis exposure and differed in, a whole bunch of things like sensation seeking, a whole bunch of mental health measurements, mental health diagnosis, ease of obtaining cannabis, peer influences, intentions to use. What do you think cannabis is going to be like when you use it? So, there's a whole bunch of features that are predictive of future cannabis use. And we can start coming up, if you will, with, a risk profile. This same group also just looked at, well, what might change once you start using cannabis? So again, very, very simple idea. They identified kids who were substance naive at 10, who went on to use, in this case, cannabis, but also drugs by age 15. And then they just went back to their 10-year-old data and said, okay, well, the future users, did their brains look different before use compared to those who didn't? And what they found, we won't go into the details, is essentially thinner prefrontal cortices and thicker cortices elsewhere in the future users. So again, we have here, for whatever it might mean, but a brain predictor of future cannabis use. And that kind of raises all sorts of interesting questions as to what that might reflect. In our analysis, and this is the work that Tony has done, and I will try to wrap up here as quickly as I can. We tackled this in a particular way. So we restricted our focus to those who initiated their use after their year 2 scan. So we wanted kids who were not using cannabis at baseline and also were not using cannabis at year 2, and then went on to use cannabis. And why did we do that? Kind of for the following reasons. If you have brain data, let's say at age 14, and it doesn't matter what you're measuring, but you compare cannabis users to controls and you see a difference, that's interesting. Maybe that's related to their cannabis use. So it is an evidence of a cannabis-related effect, but of course it is cross-sectional. So with all the qualifiers and caveats. If you have, preceding data, for example, from a couple of years earlier, and there's no difference, this is prior to their substance use, then that's kind of nice. It tells you there's no pre-existing difference. This difference you measured looks like it emerged with their cannabis use, more plausibly related to the cannabis use. But we have longitudinal data, so we can potentially even go further back in time, because there is this risk that maybe all that you're seeing here is just the, emergence of a pre-existing trajectory for whatever reason. So the idea here just basically is that it's nice to have a clean baseline pre-initiation, but it's even nicer to have a pre-initiation trajectory. So we can kind of get a feel of what the trajectory looks like, and then is there a departure from that trajectory once something like cannabis use initiates. In these data, we also can split the participants into those who used early, so relatively early between ages 12 and 14. So we have our baseline scan, we have our age 12 scan, and we've got a group who started using between 12 and 14, and then we have a scan at 14 and 16. And then we have a second group who started using between ages 14 and 16. One of the big questions here is, are there especially risky windows of time? If cannabis has an impact, does it have a bigger impact in the younger individuals? And then the other thing we can do, and Tony would be only too delighted to talk with you at length about this methodology, but I spent a big chunk of time trying to address all the confounding. Again, I think about this, a couple of slides back, I showed that there are pre-existing differences between those who go on to use cannabis and those who don't. We can't pretend that they're not. So how do you accommodate that? And so Tony adopted this approach to sort of, weight the controlled participants by how similar they were to the users to try to control for all these factors. And it's a bit busy, but there's a lot of things that are being controlled for here. Again, things like mental health symptomatology, any alcohol, tobacco use, cognitive abilities, prenatal exposures, puberty developments, and some socio-demographics. So these are all being controlled for in these analyses. So again, try to help us, if you will, see if we can zero in on a potential exposure effect. So we have early initiators. They're compared to their propensity match controls, and then late initiators. So early between 12 and 14, late between 14 and 16, also compared to propensity match controls. What do we see? Little bit busy on the top here are our early initiators. On the bottom are late initiators. So the data are, because this is what's been controlled for, I'm not showing you the baseline year 1. So I'm starting with year 2 data. So you'll see here in the analysis that in the early initiators, and just starting up here in the top right, the CBCL externalizing score, once cannabis use initiates, you see this notable increase in the cannabis users in externalizing. You see something similar in attention problems, something similar in internalizing symptoms, not so much in the BPM measure. in the late users, so again, they started using between ages 4 and 5. You see something similar for internalizing, evidence of attention problems, evidence of externalizing problems. So we see sort of an elevation in these mental health symptoms that arise after the initiation of use, and that seems to be there both in the early initiators and in the late initiators. And if we look at the brain data, we also see some differences that seem to arise at the same time as their use. Curiously, and the sample sizes aren't huge, 150 in each group, we don't see any differences emerging in the late initiators, or in this case, mid. So again, 14 to 16. But just to unpack this, so again, we have our controls, we have our users. We're not seeing any differences, but then once they start using, in this case, they started between years after year 2, we start to see differences emerge in brain. And you'll see that across a few regions. But again, like I say, curiously, not so much in the late initiators. So maybe in terms of brain, there is a sort of sensitive period here that we need to attend to. Okay, to finish up, the good-looking guy in the middle is Tony, and he's here to answer all your difficult questions. Also, a large bulk of this work has been done by my colleagues Matt Alba and Badr Chirani. Very, very, very briefly, just to kind of put a quick summary on this. The ABCD data set, it really is a resource that I think is very rich. It's available, freely available to you all once you register as a user. And as I say, it's definitely, I think, increasing in value as more and more longitudinal data accumulate, and there are more and more interesting outcome measures to be had. The task fMRI data, I think, is particular insight into the brain correlates. Again, I have to make a pitch for it. All those other measures, brain structure, diffusion measures, resting state measures, they don't actually tell you anything about brain processes. It's all an inference. You really can only get a handle on what a brain region is doing if you're probing it with a task. So that's why I think it's of particular interest for understanding development, cognition, mental health. And then finally, with regards to the cannabis, there is evidence of mental health and brain changes arising with cannabis use. So I want to be very careful not to make overly causal language, but certainly the two tend to co-develop these mental health issues, these brain changes with the cannabis use. And our goal here, just to let you know, kind of moving forward with that particular plan of attack, is with these rich data, as best we can, we'd like to move beyond the kind of association analysis and use whatever methodologies we can to arrive at maybe more causal inferences. So exploit genetic differences, exploit the twins discordance, use more of these cross-lag models. There's numerous kind of causal modeling methodologies that we're employing as well to try to get a handle on this. With that, I will stop and happily take any questions from you. Thank you for your attention. David, do you want me to stop sharing?
Yeah, why don't you stop so we can
Yeah.
see, unless someone needs to see something in response to a question. Thank you so much. That was fabulous. I think people can unmute themselves to ask questions. I will start with my prerogative question. Um, I'll phrase it this way for the fun of it. Is small science doomed in the sense that you have to scan 10,000 people in order to get future small samples? I mean, by the time you get your genetic similarities and your siblings and you covary for all the things that are you find out to be important, you still may, even in these big data sets like ABCD, end up with small, regular types of samples,
Yeah.
but with much cuter features. That makes me so anxious for everyone doing regular science. So I obligatorily have to ask,
Yeah, yeah, yeah.
what's the poor regular person to do when they're trying to do a smaller study?
Right. Good question. And I just see that my old pal JB Pauline has joined us, and that's lovely. Speaking of all things irregular, hey JB. Yeah, no, I mean, I think I'm sure you guys have discussed this at length. Not at all. I mean, I think you're right about the big samples. I mean, they're never big enough, for one thing. I mean, 12,000 subjects is nearly big enough. We need 100,000, we need a million, because once you start asking interesting questions like, I hypothesize that cannabis exposure effect is really only be there in those with a family history of, and who are female, and who have a polygenic risk score 4, and have had such and such environmental influence. That's the group I'm interested in. That 12,000 diminishes to 50 very, very
Yeah,
quickly.
yeah,
But I think I mean Whether or not you can, what you can do with small sample sizes will, of course, depend on the effect size. So if you're anticipating a large effect size, I think you can get by obviously with a much smaller. And if you're doing any kind of an intervention or a pharmacological challenge, or you're doing very dense sampling within individuals, so you can get your big data within
Yeah.
rather than between people, then I think there's all sorts of useful things that can be done with small sample sizes. I do think small samples and then between group comparison or correlations between behavior and brain in a small sample, that stuff kind of leaves me cold a little bit these days. This is the stuff, of course, with which I made my career. So, but that I think, yeah, that's a much tougher sell. But I think again, bigger effect sizes, interventions, treatments, dense sampling within people are all very useful.
Great. Instead of following that up, I'll see if anyone else from the audience can ask something. Oh yeah, Dura, are you able to unmute or did I do something? Okay, good.
No, yeah. Thank you so much for presentation. I was just wondering, because you were talking about like the cannabis studies and also like, also like how important it was for you to for the study that, you only take kids that were not using at the beginning and only later started using, etc.
Yeah.
But I was wondering, so are you relying fully on the self-assessment or like kind of
Sorry, say it again. Are we allowing?
relying only on self-report?
Yeah. Well, yes and no. So, for the most part, yes. Though we do have some biochemical verification. So we do have hair samples from all the participants, but they're not all processed, they're not all analyzed. So typically, depending on the research question, we would, for example, exclude a control, or identify a control as a control if they don't report any and they also don't show any positive, cannabis in their hair. But for the most part, I think our cannabis work, we are dependent on self-report, and it's very hard not to, because you'd need much more frequent biochemical, sampling and biochemical analysis to really confirm, and we just don't have that. So we get hair samples when the kids, come in, and they come in every year. So yeah, a lot of it is self-report. Now again, do we trust it? This is always the question. We have done some analysis where we're comparing the self-reports with the hair samples, and there is some discordance. For the most part, we do have kids. It tends to be in one direction, and correct me if I'm misrepresenting this, Tony, that kids who we rarely have kids who we often we have kids who underreport, so they tell us they didn't use cannabis, but the hair samples say otherwise. So that does happen, and that's why if you
That
have
would be my guess, yeah.
yeah, but yeah, so that's where it tends to occur. And I'm sorry, this is a much longer answer than you probably wanted, but as best we can, we try to explain very carefully to the kids that their data is fully confidential. We will not tell their parents. We try to make it very, non-stigmatizing. They know that a lot of their peers, etc., are using. But for sure, there's going to be a little bit of variance within there with the
Okay,
misreport.
but it looks like you also like sometimes studies the hairs and sort of.
Yeah. And some people, for some analyses, will restrict their sample just to those with the hair positives. And
Mhm.
that would obviously be great. But again, and I'm sorry, I'm rattling on, but that just gives you a sample in time. If it's self-report,
Yeah,
you can get these timeline follow back. So if you believe the self-reports, you can get then better,
yeah,
frequency information, better, yeah, dose information.
yeah. Thank you.
Sure. JB.
Yeah. I was sorry if that's a little bit of a technical questions. I was curious about the twin analysis. I think I've seen a couple of papers now on that sort of things. Every time I kind of ask myself how are so usually it's a Gabor distance that is being used between two individuals. and it's never always clear to me how the weights of that distance are kind of learned. Do you have do you know a bit of what how that was done, or maybe sorry if that's too technical. I
No,
mean
no,
that's
no.
okay, but okay.
Yeah, I think the way it is simply a difference in, let's say if it's in the one example I gave was screen media activity, number of hours spent playing games.
Yeah.
Uh, it's simply just the number of hours and then the difference score between the two. And then similarly with the mental health measure.
But you're not aggregating a number of characteristic of the sum of the subjects. Like there's a maybe a number of hours for the game, maybe some genetic distance, maybe like
Yeah.
some to find the closest cluster of individuals. I mean, it's not only on one dimension, right? It's on various
Well, we,
assessments that are possibly like binary, not binary, or qualitative and quantitative, and so it's a matter of how do you what does it mean to be the closest individual to another one? Like, is
Yeah, so for sure, and I'm not sure if this is exactly what you're asking. The analyses do include a number of, covariates that we want to control for. I will say, if you are interested, because I suspect I probably can't give you a satisfactory answer right now, but the guys who worked on this here, there were two in particular, and they spent a good year working on the analytic method, because these models are actually quite complicated
right.
because you have random intercepts. You've got some trait-like variables that you need to control for, but then within each of the cross lags, you've got variables that are time variant, and you want to accommodate for them. And one of them is a bona fide mathematician, and the other is one of the smart, a really sharp psychiatrist, and they really went to town getting into the weeds of the analytics. And as you probably know from these models, getting them to converge and getting robust measurements is really challenging. So if um, because I know you probably, knowing you, I know you are very interested in the particulars. Happily will set you up with those guys if you want to talk through the mechanics of it.
I'd be interested. I'd be
Yeah,
curious.
yeah, I
It's
know. It's
like
complicated.
it's complicated because the closeness of two individuals
Yeah,
on a multidimensional, like different assessments, different characteristics. To me, so kind of depends on what is the closeness going to be used for. So it has to be so the weights on the characteristic of the samples probably have to be learned with an intent. And so that's why I mean like yeah. Anyways, I think
yeah,
that's but it was lovely work, super
yeah,
work.
yeah, yeah. No, I would. They'll happily chat with you. I cannot tell you the number of meetings we had where we really got into the weeds of the analytics. So they would love to have that conversation with you if you want.
Thanks so much for the presentation. That
Yeah,
was
yeah,
super.
yeah. Good to see you.
And I left in the chat a quick paper that Michelle can talk about in detail that you might be interested just to look at some stage. It's I mean resonating with what you've said.
Ah, okay.
Great. We'll make sure that gets shared if that went by too quickly or maybe Tanya grabbed it. We are at the top of the hour. I thank everyone for attending. I thank Hugh for presenting. I thank all of this is the last of our current academic year presentation, so we've ended on a very high note. That's great. We will start again. We're not going to do September. We'll start again in October after the holidays. So we hope everyone will pay attention and join NICAN next year. Have a safe summer. Have a safe travel to OHBM or other conferences if you are doing that. And we will see you all in the next season. Thank you all.